Take it away.
Hey, thank you, Peter, for that wonderful introduction. And thank you, everyone. I just need to make sure everyone has a card. Does everyone have a card? And someone just came in. So where's the deck? Oh, yeah. Okay, so brief apologies to those on Zoom. I can't get the cards through the Zoom. So for those of you in person, we just have a little interactive piece, and I will try very hard to stay here in front of the computer.
Your microphone, yes, please.
Because I tend to wander when I talk. Okay, so thank you everyone for joining. And as Peter mentioned, I'm going to go through the work that's been accomplished so far for my master's thesis. And the title I have for that today is Pathway to Personalized Medicine, Evaluating One Single Integrated Method for the Molecular Profiling of Diverse Driving Alterations Across the Spectrum of Myeloid Neoplasms. So to dig in here, as an overview of what we're going to be discussing today, briefly, I will go through the driver complexity that exists in myeloid neoplasms, and also a little brief history on the successes we've made so far in personalized medicine in our industry, and how that translates to the current clinical problem that we have in the hospitals and in the laboratories. That will lead into my hypothesis for my project and the associated objectives, followed by the experimental approach that I've undertaken, a brief look at the validation results from this work, and into the clinical efficacy and efficiency data. And in the end, the future directions, because no problem is ever solved all in one go. So overall, our blood system, just briefly, is comprised of a multitude of cells. They all have various functions. They all work very hard every day to keep us alive and keep our blood flowing, clotting when it needs to, transporting oxygen, all kinds of things. So I'm not going to go into the details of all of these cells in particular, but this slide is just to highlight that there are a lot of players involved in our blood system that all are required to do their job to keep us healthy. So when things go wrong, there is a continuum of hematopoietic disorders that can arise because there are so many cells at play. And so what can happen is, from a normal state, we can transition into myeloproliferative neoplasm, or an MPN, which results from an increase in proliferation. So we have a lot of cells being made that are generally still of normal morphology, but there's just way too many being made and they clog the system and cause disease that way. Another way is we can shift from a normal state to a myelodysplastic syndrome, which is where we have not necessarily an increased proliferation, but dysplasia or abnormalities happening. So those cells, they don't look right, they're not functioning right, they're not able to differentiate, or they're not able to mature properly. And so there's also an overlap here of an MDS and MPN syndrome together. And that occurs when you have both the high levels of proliferation and dysplasia in at least one myeloid lineage. And so either of these three disease states can evolve into an acute myeloid leukemia, or an AML. And that's known as a secondary AML because it is secondary to their initial oncologic condition. And so that usually happens as the disease progresses, as there's an additional layer of loss of differentiation and just the growth of all the cells. AML can also occur from, a somewhat normal state, meaning the person's they don't think they're actively sick. And then all of a sudden they have problems where they present into the ER and they do blood work and there's indications of an acute myeloid leukemia. So when we look at how these cancers develop, they develop from drivers. So when I say what driver, what I mean is a change in the DNA sequence that's basically causing this cancer to develop. So this can be either a driver from germline origin, meaning a variant or a change that you were born with, or acquired somatically through your lifetime. And this figure here is specific to the MDS and MPN overlap. So just to clarify that, but what it's trying to show here, the takeaway is that these drivers usually start in one gene, and then you get an accumulation of hits. So you have various changes that occur, and they hit certain genes that are involved in either epigenetic regulation, so with methylation, or with splicing, or with increased proliferation for KRAS and NRAS, for example. And the individual profile that results, so that person's condition that they have, is quite personal to them based on the timing of these variants, the sequence that they develop, and how their cancer develops. So when we look at drivers, there are, generally speaking, 4 different types of changes that happen in the genetic code. So there can be what's called a single nucleotide variant. What that means is just one nucleotide is changed to be the wrong one. That can generally happen during a replication error, or from other types of ionizing radiation or UV exposure. A multinucleotide variant, so an MNV, what that means is it's when there's a stretch of nucleotides in a row that are abnormal in some way. So that can be either an insertion of a little foreign sequence, so that sequence isn't there normally. It can be a duplication of a little bit of a sequence, or it can be both. It can be really fun. So it can be like that. Now, as long as it's less than a full exon, it's called an MNV. Once it's an exon level, so if it's a full exon that's deleted or duplicated, generally speaking, we refer to it as a copy number variant. Copy number variants can also be larger than just one exon. So it can be 1 exon, 4 exons, the whole gene. And then we also have translocations. So that's where a piece of a chromosome, usually it's at the chromosomal level, but basically one chunk of DNA moved to go to another chunk of DNA. And that you can see at the chromosomal level because it's generally large stretches of the sequence. So not only do we have these different types of changes that are present in the DNA, we also have them happening in genes, which I touched on this a little bit earlier, but it's happening in genes that are involved in a variety of processes in our body and in our blood system, particular for this discussion today. So when we're talking about myeloid leukemias, we can have variants that occur in TP53, for example, that impacts the apoptosis pathway. We can have DNMT3A or TET2, which does the methylation, or ASSL1 or EZH2, which are involved in, this is particular here is showing the chromatin packing of those chromosomes. We can have abnormalities in RUNX1, CBP alpha, transcription factors, growth factors like KRAS and NRAS, genes that are involved in the chromatid. So this is when the chromosomes are lining up to do their dividing. So STAG2 is a player there. And also splicing factors like SRSF2 and SF3B1. So there's many different genes that I just named a few that can be involved across these different pathways. So, well, what does that mean for our work? So our work that we do in research is we try to identify, well, can we come up with what's driving all these cancers? The work that I'm doing is blood cancer. There's lots of other important work everyone else is doing as well. Can we find a variant or a gene or a transcription level, something that can help us develop a treatment? So the most, or the oldest example of that, I was going to say classic example, is the first consistent chromosome abnormality that was identified with cancer was discovered in 1960. And it is this translocation between chromosome 9. So this is a normal chromosome 9 here on the left-hand side. And this is a normal chromosome 22 on the right-hand side. And so what happens in this type of CML is you have the bottom dark band of this chromosome 22 and the peripheral fluff there at the end translocated onto the bottom of this chromosome 9. So what ends up happening is you have a derivative 9 chromosome, which is here, which almost looks like it has three dark bands, although one is significantly less intensity-wise than the other two. And then you have this little derivative 22 chromosome, which kind of looks like a little kernel of popcorn down the mic. And it's known as a Philadelphia chromosome. So this was discovered quite a while ago in chronic myelogenous leukemia, is specific for this abnormality. And what happened was we were able to develop a kinase inhibitor that directly targets this fusion product specifically. It's known as imatinib. And the first clinical trial was in the late 90s. And so one of the studies that I looked at, which looked at the first 10-year period of it being in use, had an 83% disease-free survival. And I believe it's gotten even better since then. So this is one of the older examples. But since then, we've also been able to identify JAK2 variants in myeloproliferative neoplasms to discover a JAK2 inhibitor in 2011. And for AML, so acute myelogenous leukemia, there's been the discovery of a FLT3 inhibitor, which is specific, in this figure here, specific to the tyrosine kinase domain activation variant that can happen. And there's also been more recently IDH1 inhibitor in 2022. So, I mean, all of this to say that we need to detect these variants and we need to see what's driving the cancer in order to be able to develop an associated treatment. So when we look at, well, what's the clinical problem? How does this actually, where's the pain point here? Right now, because there are so many genes at play and there are so many different types of variants that an individual can have, traditionally, we've been using multiple targeted tests in the hospitals in order to diagnose someone's cancer fully. And it's almost like, I mean, I don't want to, not fully a fishing expedition, although there is a fish test and I'm not trying to be funny there, but so generally speaking, you order a bucket of tests and you're going through all of those results and you're pulling all of those together. And this talk is not to say that those are not important. Those still are important. However, I mean, they're important because they're rapid, generally speaking. They are tied to known and precise therapeutics. They generally have a low cost. They are limited in scope, though. So when you look at these targeted tests, you know, it's real-time PCR for JAK2, and it'll detect V617F, but it won't see other things, which that's great if you have that variant and you're detecting it. But if you don't have it, then that's not so great for that person in the sense of the testing, I mean. And so the idea was, well, what if we looked at a different strategy? What if we looked at a comprehensive sequencing strategy is the terminology I'm using here. And what I mean by that is instead of running multiple targeted tests that are only examining one particular change, one or two, what if we did a broader sequencing approach, not as broad as whole genome, that's not what this is, but a broader approach with intention and design to detect both the drivers that we are supposed to be looking for, but also potentially novel drivers, right? So we want to be able to find some changes that occur that maybe we didn't know about before. And this type of method, yes, it'll cost, there'll be a sequencer we need, there'll be data storage, there'll be analysis requirements, but we'll also be able to have an in-depth look at these patients, potentially stratify them for risk and develop different prognosis metrics, and also incorporate copy number calling in that as well. So essentially what we're trying to do is translate all of this information that we can achieve from a comprehensive sequencing strategy and translate that in order to improve patient outcomes. So my hypothesis that I've had for my master's work here is that a targeted massively parallel sequencing assay, otherwise known as NGS, with broader gene and DNA copy number coverage will enable the detection of multiple drivers of leukemia and for myeloid neoplasms with increased efficiency and efficacy. So to examine that hypothesis, I laid out two objectives. Objective number one is to design and clinically validate this said assay for multiple types of drivers. And objective number two is to develop technical and clinical standards for the interpretation and reporting of these variants. So as a little refresher, this is the same slide from last time, but essentially this work encompasses 4 stages. Stage 1 is the assay development, and we came up with a name with the help of my friends in the lab of SMILEY. So SMILEY stands for the Somatic Myeloid Lymphoid Panel. And stage 2 and stage 3 encompass the assay validation. And leading into stage 4 is implementing the assay and then being able to finish the rest of the metrics here. So I'm currently in stage 4 with respect to the work right now. But what I'd like to do is just quickly walk through that assay design so that everyone is on the same page. So when we looked at designing this assay, we picked out 49 genes to be fully sequenced, which means all of the exons are included in the bed file or in the region of targets for this panel. And then another 42 genes that are there for hotspot coverage, which means just certain exons of interest. We included known myeloid and lymphoid targets that have lots of literature on them, and we know that they have pathogenic interest, and also a few targets for germline interrogation. So overall, this panel is an additional 51 genes than the previous assay in use, and it's actually 91 genes in total. In addition, we did the copy number coverage. So this is a figure, an ideogram of the chromosomes in the human genome. The horizontal lines indicate the location of the genes of interest. The vertical lines indicate which chromosomes we included backbone padding for to make sure we had enough coverage of those chromosomes to identify if there was a large chunk of them missing or duplicated. So we specifically, with intention, selected certain chromosomes for this purpose. So we have chromosome 5, 7, 8, 9, 11q, 12, 13, 17, 20, X, and Y. And it turns out, incidentally, we actually have pretty good coverage of 21 as well because of the genes that are on there. And so we are able to see duplications of 21 as well, but we didn't intentionally pad that with respect to the design. So when we went to choose our method, so I did not put in the zebrafish, David, I forgot. So this is now, last time I had a zebrafish here. So just so you know, this is now a bone marrow, I fixed it. So this is the method that we decided on, which was the hybrid capture sequencing method. So essentially, we have leftover clinical samples that we used for the validation, and we extracted them by 3 different methods in total that all were either column-based or salt-based extraction methods. After we extracted our material, we did quality control, which is a fancy word in this case for saying we measured the quantity of the DNA that we extracted, and we made sure that the DNA was intact, so it wasn't too fragmented to continue with the work. We then did library preparation for hybrid capture, which means we did an enzymatic fragmentation method to chop the DNA sequences up. We then continued to use an enzymatic method to add on the adapter sequences on the ends, various bead cleanups in there as well. And then we quantify the libraries that we obtained, and we set up our hybridizations in 8 patient samples per probe pool. And that's important because of the probe ratio to the amount of library. And the sequencing run can actually fit 4 of those pools of 8, which means you have 32 samples on one run of this particular sequencing chemistry. And this sequencing run takes about 17 hours, so it runs overnight. And then once it's done, the OGT interpret software is what we've been using for our analysis. There's a few hours of data transfer and data alignment in the software. And so this interpret software produces the results for copy number and for the SNV and MNV variant detection. So when we look at the validation itself, there was 3 runs in total with 32 samples per run. This side of the table here represents 65 unique patients that we included in our validation. And we wanted to make sure we had a representation from various types of clinical phenotypes and disease subtypes for the myeloid neoplasms. And that's represented here in this column. This chart on the left-hand side is a breakdown of how many unique patient samples we had, how many we reproduced. We also purchased some synthetic control samples that are ones that you can buy commercially that have known profiles in them. And we also included population samples, and this was important because we wanted to be sure, because we're dealing with copy number for the analysis, we wanted to be sure we had an understanding of what a variance of normal looks like in individuals being sequenced by this assay. So we had that as well. And we also reproduced 6 of those as well for more data. And we did a limit of detection and a limit of blank for the validation. So overall, there was an average total mapped reads per sample. Sorry, this isn't mapped reads, this is total reads. 6.9 million was the total read output per sample on average. And the mean coverage was just over 1,000 mapped reads here per specimen. And we did have some libraries fail. So our failure rate was around 5% for the validation. It's gotten significantly better since then. And these failures were particularly due to, we had a few that had shifted insert sizes, meaning when you're doing the library prep, there's a bead cleanup that is also functions as a size selection. You want to make sure your libraries are all, in this case, 325 base pairs, but we had some that were too small. So something went wrong with the bead cleanup. And so those ones failed. And we also had a couple that failed for coverage. So even though everything looked good all the way along, when it was sequenced on the sequencer, the coverage didn't meet the minimum. Our minimum is 500 mapped reads to do proper analysis for these samples. So this is a graph of our, a couple other metrics. I won't go into too many. This is the duplicated read rate in the orange bars, and the on-target rate is the gray line. This red line represents the threshold for duplicated reads. You ideally would like your duplicated reads for NGS to be much lower than 40%. We had quite a tight run for run 1. Run 3 has a little bit more wobble there, but overall, all of our runs were good and under the 40% cutoff. Duplicated reads can happen when you have errors in, so if you have too low of an input and you're trying to not compromise, well, sort of compromise for that by amping up the cycles. If you have a PCR step in your process, you can incidentally increase your duplicated read count, which can be something that happens. Also, if there's any type of bead cleanup issue with the ratio of beads to library, you can have that as well. So, but overall, we were good there. This is a graph, not to overwhelm. So sorry, I didn't put on a definition on the axis because it was too many words, but essentially each line that you see is a variant. The orange dot represents our variant allele fraction that we obtained with the SmileyP panel. And the blue dot is the variant allele fraction that was obtained previously on our other technology, which was semiconductor-based, amplicon-based OMP. So this graph isn't to say anyone is right or wrong. This graph is simply to say that there is variance in how methods detect the presence of a driver, and also the fact that every sample that we ran, meaning our unknown sample, like our known patient samples that we ran, were all concordant. So all the variance that we saw by our previous method was still seen on SmileyP. And SmileyP was able to identify additional ones, which I'll get to in a few slides. So for clarity here, there were a handful of variants that were discarded due to them being technical artifacts. And the definitions for that we used were variants that had a greater than 80% alternate allele strand bias or less than 2% VAF when they're in close proximity to a homopolymer region. And a homopolymer is defined as more than 3 or more of the same nucleotide in a row. So this, if we hone in now on just a few variants from that previous slide, these are 4 particular variants from that larger data set that are variants of known drivers that are interesting. This is a duplication, this is an insertion in NPM1 that's quite well characterized, STEG2 SNV, and an ASXL1 deletion. And so each assay saw all of these. Like I said, everything was concordant. And this is just to demonstrate the differences in their variant allele fraction. And Again, this is not to say anyone is right or wrong. It's just to highlight that methods inherently do function slightly differently in their ability to translate the presence of that biological variant into an output of here's how prevalent that variant is. So this here is an overview of the entire validation. So we had 78.5% of cases that had driving variants in multiple SNV and MNV type of composition. So they were either multiple nucleotides or a single one. For our sensitivity and specificity for that detection was 98% and 100%. Now for our copy number, which is represented down here with the larger rectangles, the SNV and MNVs are up here with the smaller squares. Our copy number had a little bit less, which we were expecting for sensitivity and specificity. And we actually had a false negative rate in our CNVs was a little bit higher than we would have liked for our validation. And so we found that if there was a clone that was less than 50%, we were missing it at first with this technology. And so what we did as part of our validation process is we went back to the vendor and had conversations and were able to tweak the algorithm so that we could further reduce that, meaning now we can see down to 30% clonality. And there's still more work to be done there as well. But that's just an overview of that. Sorry, I guess I should say, yeah.
Question.
Yeah.
Maybe I missed it. What's the thing you gotta get? What's your gold standard?
Oh, I'm getting to it, yeah.
Oh.
But yeah, so you're right. When we do a validation right now, the samples are being compared to the previous method, but that's not a true synthetic sample. That's what you're asking about, right?
No.
Like a characterized one?
No.
Yeah, so generally speaking, in validations, you look at both samples that you've run on other orthogonal methods, but then you also do a synthetic control. Yeah. Oh, the orthogonal method is OMP on semiconductor sequencing. Some of this, for the copy number, we looked at karyotyping, yes, and fish as well. Yeah. Yeah. So, and this, I forgot to mention, this bar at the top here is dividing. This is just indicating the disease subtype of our patients. So there was almost half of our patients here were AML. And then we had a handful of CML, MDS, and MPN as well for the validation. So this meant we were able to finish our validation in the summer of 2024. And then we began our clinical implementation in right after that. And so we have about 10 plus runs, I think more than that now. But for the purposes of the next few slides, I'm going into the first 145 unique patients that we processed in the clinical lab. So this was our incoming case volume divided by disease subtype. Based on the referrals that we were received. And for the purposes of this presentation, I have created some lumping categories just because we didn't actually divide out a lot of them. So other, for example, has mastocytosis, anemia, and eosinophilia in there as well. Oftentimes when we receive, not often, but sometimes when we receive samples for the SmileyP referral, they haven't quite figured out the exact, whether it fits into MDS or MPN yet. So this is we had 27% of MDS. That was the highest level, as you can see there. And when we look at the library yield, so this is the total nanograms we were able to obtain for each processed library. Our minimum threshold was 100. Everyone's above that. And you can tell just kind of visually, as these are plotted in the order they were processed, there is some slight batch effects that can happen. For example, right here with a slight drop, but everything was still good and over 100. And this is our mean coverage for those same set of samples. And this is representing, the red line here is representing our threshold for acceptable coverage, meaning 500. There was a couple samples that failed this threshold that we were not able to do analysis for and had to be repeated. So overall, when we look at the results, for 54% of the cases that we did, and this is just for the first 10 runs, like I said before, 54% of patients had what I'm referring to as a combination detected result. What I mean by that is they not only had SNVs or MNVs detected, but they also had copy number changes as well. And we were able to identify all of those in the one test. There was still 13% that were not detected. And there was still a large amount of cases that were just SNV only. And so this does still speak to the fact that there are still a lot of cases that you could detect doing the multiple standard test way, although we could get into the different genes these are all in and how extensive that would be for working up all of those various gene targets. But in essence, this was really happy. We were really happy with this, that there's over half that we could get the combination detected. So this is looking at the SeraCare synthetic control. So this is comparing the SeraCare control has digital droplet PCR measured variant allele fractions for 18 relevant myeloid variants of interest. And some of these are deletions, duplications, insertions. So there's a diverse array there. The black square is the expected variant allele fraction. The green diamond is what we saw on our Smiley panel, which is produced by OGT. And the red triangle is what we previously saw with OMP or the semiconductor technology. The semiconductor technology does have some known areas that it struggles with, mostly CBP alpha because of the GC-rich area that's there. It's not able to identify the single base pair insertion that happens there. This one here, it missed in this data, and I should reiterate, this data is just one dot on a line, meaning this was just one time that we ran the SeraCare control. In the clinical lab, we do run it multiple times. I just took one data point for this purpose, but there is variance that you do see when you run a control over and over again, batch effects and other reasons. So variance is expected, but this is the absence of the detection of these three calls here. And this is the FLIT insertion specific to that. There is a workaround in the analysis where you can, for thermo I'm speaking about now, you can pull this up, but it requires a special separate pipeline. So in any case, we compared this to make sure that, yes, we're able to see all of these variants properly by our Smiley assay. We knew before we had all of the variants concordant from the patients. This is to make sure that for the synthetic control, we were detecting all of that. And so this is a look at our, I suppose, hit rate. So the proportion of this is specific to pathogenic calls by gene and variant type. So when we call something pathogenic, what we mean is there's enough evidence in the literature and functionally speaking to say that it has clinical relevance to drive that cancer. So we had 369 total variants that fit the pathogenic, likely pathogenic bin from the first 145 patients that we examined. And our highest hitter was TET2, which is what a lot of others have also found, in addition to ASXL1 as well. Our third hitter was SRSF2, which was interesting to me, the splicing factor there. It has a role in splicing, sorry, is what I mean. And then when you look through, you can see that there was a lot of the common genes that are involved in either differentiation or growth, like KRAS. And for copy numbers, there was the del7s, so del7p, del7q, a gain of 8, meaning a duplication or an additional presence of the entire chromosome 8. So overall, we had 52 deletion variants, 28 insertions, 11 MNV. And when I say MNV here, what I mean is the internal tandem duplications or ones that are both an insertion deletion together. So when we looked at our efficiency, what we wanted to see was, well, okay, we can see all of these drivers, but how does that actually translate to the work that we're doing with respect to the diagnosis that we're making for these patients? So the green bars here represent the number of patients in each of these disease subtypes. And the blue bar on the left is the total number of reportable variants for that cohort by disease subtype. So we had 32 AML patients, and we reported overall 187. And the bar in the middle is representing how many tests this would have been if we had done everything separately, based on the copy number being a separate test from the SNB and MNB. And so when we do the math of sort of, it's kind of rough math, but with respect to the efficiency, what we're seeing is for every one patient of AML, we needed 2.3 tests the old way, let's say. Whereas with this one assay, it's just 1 for 1. So it's the 32 patients, 32 tests. And so what you see is the biggest impact is actually with AML and the leukemias over here. And this is a bundled group of multiple different types of leukemias in here, which we're going to work on separating out that data now. But also interestingly, what you see is the MPN subtype doesn't have as much of a win, let's say, for doing this assay. It was almost equivalent for that. So now I thought what I would do is just walk through some of our most interesting cases. I have 4, I believe, just to look through and see, to show you with some pictures. So this is a discovery, or we were able to identify isochromosome 17q. So what you can see here, this is the IGV plot. The bottom section is the loss of heterozygosity. And this section here is the copy number variant calling. So this is the normalized ratio values. 0 is the line here. So this result showed us that 17p in the location where TP53 is was entirely deleted from this sequencing. They did have another copy of a normal 17 though, because as you look down here, the loss of heterozygosity shows you that all the variants were either at 100% or near 0. And then this 17p11.2 region is showing duplicated, which in the literature, there's actually, someone has already discovered this before, and they call it a cryptic duplication of that P region because cytogenetically you can't visualize this because it's so small. This part, I mean. And then here is the 17q. So to look at the pictures on this side, this is a normal 17 on the left-hand side, and this is a IsoQ, a 17q. So it's the entire Q arms, 2 of them stuck together, which you can easily see in the cytogenetics there. So this was super exciting that we could identify this on the molecular level. This one was it was a TALL case, actually, and it was a young individual, like a young adult age. And we saw in our copy number calling a deletion that was the entire coding sequence of DNMT3A. Now, we weren't able to show or see if that deletion extended further on either side because we only had DNMT3A in there. We didn't pad chromosome 2. So this is all that we're able to see in the molecular lens that we had. But this individual also had a 9p and a 12p deletion. And then when you looked at the SNV variant profile for this person, they had 2 Notch1 variants and a splicing variant in PHF6, which was very interesting because there is literature on the fact that in cases of lymphocytic leukemia, the Notch and the PHF6 are known to coexist together, and they sort of help to drive the development of that cancer syndrome. So that was also very cool that we were able to see all of that picture together. This is one where it was a case of an individual who had MPN. So they had a JAK2 V617F for some time. And then they were getting worse in their disease and getting sicker. And so we tested them again. And then they had a del5Q, del9Q, del13Q, like clear cut. This is just a picture here of an example of what that would look like. This isn't this person's chromosomes, but this is a normal 13, and this is the del13 type that they would have had. And the plots here are for the copy number as well. But they also had two variants in TP53. So they had the serine 215 asparagine, and they had this super interesting extension variant. So essentially, this was an insertion that happened in the second last codon of TP53 that what the implication was is it actually extends the protein instead of truncating it. And when you look up that variant in the literature, there is nothing that we could find on that particular variant, but there are other stop loss variants that are noted in TP53, which is a whole other talk that I could do. And then this one here also was super cool because we had an AML, so an individual that came in with query AML. The diagnostic marrow found a handful of things. So we found the NPM1, we found the SRSF2, the IDH1, and the RUNX1 frameshift. And there was also this GATA2 single nucleotide change here. And the variant distribution, so the frequency of each of these drivers was different. They weren't all the same. But, that's not entirely uncommon, but also we can't really say anything about the status of any of those when we're doing the diagnostic marrow. But after the individual went through treatment, we did a post-induction marrow and tested that one again, and everything dropped off except for the GATA2. So that leads us to the suspicion that possibly this individual has a GATA2 germline variant, and that sort of led them to the susceptibility of developing this AML in this case. So that also was very exciting because we need to validate this, but hopefully we can use this test also to confirm germline, and maybe we could also use it for disease monitoring of these individuals. So overall, I guess my takeaway points are that what our work has shown is that the Smiley P panel, it increases our test efficiency. So we're able to see more with less number of tests. We can see complex variants that were missed by the other assay before. We can confirm molecular remission status, meaning after treatment, we can see did the drivers go away or not. And all of this means that we can put all this information into a comprehensive patient result to be interrogated by the clinicians and all of that sort of thing, so they can have a full picture on this patient's cancer. So now you're probably all wondering, why am I holding this card? Because you didn't talk about these cards. So, okay, I'd like everyone to look at their card. And if you have the number 8, 9, 10, or Jack, I need you to raise your hand. Okay, so that is all of the SNVs that we were able to see in this assay. Who has a queen and king? Keep your hands up, though. Keep your hands up. Okay, look, so now we've got more hands. So now these are all the MNVs. Okay, so who has an ace? Okay, so everyone put your hands down except for the aces. All right, so these are those tricky little translocations that we cannot see by this test. So yes, we're able to detect a lot more than we used to, but there are still things that we can't see by this method. So overall, the future directions for this work is to look at honing some of this data to develop maybe some risk stratification models. I say maybe because there's other ones that have already been developed, so we'll see if we can add to the value there. And then develop clinical frameworks, look at the targets for MRD monitoring, and also go into more of the transcriptome work to examine more of those fusions and those translocations that we're not seeing currently. And so in summary, I would like to thank my supervisors, Dr. Graham Quest and Dr. Michael Raw, for all of their support, my supervisory committee as well, Dr. Janique Deba and Dr. Catherine Trishkin, the Queen's faculty and staff, so Dr. Peter Greer for organizing this, Mark and Kevin. I know Kevin's retired, but still thanks. And the two vendor reps there from Oxford Gene Technologies, my Raw Lab colleagues who helped me with the colors because I needed that help. And also the clinical lab as well, which this project wouldn't have been possible without their help as well. So thank you, everyone. I have time for questions, I believe.
Eager to take questions, but the water crowds back at the end.
Oh, yeah.
So those of you on the Zoom, if you have a question, you use the raise hand function. I'll keep an eye out for that. Questions, go ahead. Dr. Mueller. So I'm a big fan of pattern sequencing like this approach. I think it does deal with a lot of throughput issues and the like. But the trend really is whole genome sequencing.
Yeah.
As prices have come down, there's really going to move towards whole genome sequencing.
Yep.
Can you just give us your opinion about the balance between those things in terms of the advantage of this type of assay versus that of whole genome?
Yeah, for sure. So the advantages of whole genome are just what you said, that you get all the data, you get all the information. However, that's also the disadvantage. You have so much information that the data is very tricky to store and to process and to get through. So what I would say is ultimately, yes, we're going there, but much like how there's still utility for the real-time PCRs and the rapid standalones for certain reasons, there's going to be utility for those medium-sized panels that are able to process broader. So like, again, we're not getting everything, but we're getting more of the hits than with the standalone. And also the speed. Like a whole genome run takes 49 hours on the sequencer alone, right? So this is much faster than that from start to finish. So I think we'll get to a point where whole genome is definitely more in use, but we're just starting on that horizon now to gather that data and get a familiarity with it until the computing power gets a lot faster. Yeah.
Go ahead and follow up on that. What about small data and whole genome, like depth of coverage or the trade-off of?
So yeah, depth of coverage is tricky on whole genome. I am one who believes you need at least, I don't just believe it, I've seen it, but you need more than 100 coverage to get a reliable detection of these low-level drivers. And a lot of this whole genome approach that's being talked about is low pass, meaning 25, 30 coverage on average. So that means some of these genes you get 2. So it's a coin toss, right? Essentially. So that's fine for screening and other purposes like that. It's fine if you have a really high, an individual who, if we're talking about cancer, who has a high clonality level, but you're not going to find those small little variants that might be passenger variants or that might be triggering from the shadows. Yeah.
You mentioned.
Oh, so that was for copy number. Yeah. Yeah. For that, we have 3%.
3%.
Yes. 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 Yeah, and we're hoping to get the copy number improved. So we had 50% at first, then we got it to 30 with the rest of the validation. We're working on strategies to improve that further with the vendor. A lot of it is the algorithms with respect to the alignment of all the data in the back end. Yeah. Great questions.
Go ahead, Ty.
I was wondering, from those 4 case studies that you showed, are you planning to alter your panel based off of the results? I forget the one gene. There's one where you don't know what comes before or after, but even or anything for that.
Oh, for which case study? Oh yeah. So you're right. We actually are so we're doing a redesign of the panel to sort of address both of your questions for Chris and Ty. So yes, we're going to be adding in more tiling and more padding in order to have more coverage for copy number. So yeah, we couldn't see how far this deletion extended because we didn't have coverage on either side. We did have cytogenetics though, which was normal. So that is reason to believe that if there is a bigger deletion here, it's not so big that it's an entire chromosome arm. Yeah. But yes, we are redesigning it.
Questions pretty cool. Can you go back to slide 15, 16? I have a question.
Sure.
So
I love unrelated questions. Is that what you mean?
We'll see.
Okay. 15 or 16. I'm sorry. It's like, it's not letting me do it fast. Yeah. Sure. Yes.
Mean 1040 map reads.
Yes.
What does that mean?
Okay. So what that means is, Dr. Lillicrap's question, sorry, I haven't been repeating the questions, is asking what the mean coverage terminology means for this slide that's on the screen now. So mean coverage means we have all your sequencing reads when you run your test. And a lot of those reads, they first go through, I would say, integrity cleaning with respect to the sequencer itself. So a lot of this is all fluorescent based. So the light signals are cleaned and a lot of things get removed at that point. Then it gets aligned for signal conversion. So it converts those signals to your FASTQ files. And then there's an alignment to the BAM, right? So now we have our FASTQs and we align it. So mean coverage at this point is how much of all of those reads, once you do all of that trimming, is actually mapping to the area you care about. Because with a lot of these, because this is a targeted panel, and even with whole genome too, though, when you're doing analysis, you give it a list of here's these areas that we want to look at. So that's particularly what it's speaking to there.
So again, that number,
So the
that's what 40 is.
yes.
I don't have a sense. Yeah.
So we have 91 genes. So we have 6.9 million reads overall. Okay. So we got 6.9 million apples. And then we throw them at the wall or the bed file in this case. And there's some proportion that stick. So they align. And then it's the average across all those 91 genes that gives you this number. So some genes will be less than 1,000. Some will be higher.
Laurie, you could use depth as well.
Yeah, so depth, when we talk about depth, we're talking about the variant itself usually, with respect to that coverage at that depth, so at that variant position.
So that's the number of sequence read around
A chromosomal location, yes, yes. So when we say coverage and depth, that's the difference. Really, they're the same thing, it's just looking at them a little bit differently. Yes, yes, yes.
The exception or even driver variant. I mean, you've got
Yes.
the driver in your title.
Yes,
So I'm going to defend that. And
sure.
because some of these definitely involved in chip cases, right? And so there are early drivers, but some of these other changes.
Yes. So when I use the word driver, my intention of that word is variants that we've assigned a pathogenicity class of being pathogenic. So there's enough literature evidence from other cases that proves that those variants actually influence the disease to progress. So most of the data here I trimmed down into just the pathogenic and likely pathogenic calls. There's been over 3,000 variants from this work so far. A lot of them are VUSs, meaning we don't quite have enough evidence that they're actually driving. So if that's the case, they're in the VUS bin. We still report VUSs in a different section on the report so that the clinicians are aware, because oftentimes as we do research and as we gain information on these VUSs, we can actually prove, oh, here is how it drives. So that would be yeah, Chris.
You're gonna follow up on
Yeah.
David's question?
Yeah.
So it brings up the point, we are, if people are doing mutation-based sequencing,
Yeah.
for tumor, with biological tumors.
Absolutely.
They're a good chip.
Yeah. Yeah.
So but you're really looking at that.
Yeah.
Really chip, then it's not a root chip. It's extra mutation then cause genesis. Or
In some cases, yes. Yeah. Yep.
So mutations that have been in cancers. Because it's very much the same spectrum of mutations.
Right. So in the individuals where these chip variants are present at, when we say chip, we mean low levels. And so, yes, those individuals have that predisposition, and yes, they can evolve, like you're saying, into an AML state. And so I don't know that that precludes them from being the driver, though, because if in this case, these are actively sick individuals that we're doing this testing on that have come to the hospital for a reason. And so, yes, that variant could have been there at 1.5% a while ago as a low level chip variant, but now if it's 30, 15, 20, it is now part of the story.
Right.
Yeah.
So it's really.
Yeah. Yeah. You want to say?
Yeah.
Oh, yeah. With the, how do I do this faster? I can't go through this. Yeah, that one was super interesting.
This is bulk sequencing.
Yes.
So right here.
Yeah.
So look at that. Maybe you're that's the whole thing. There was also the galaxy in germline. And then what about MPM1s? You see that in MDS at all? Like where it's MPM1s into specifically? And yeah.
The AML. Yeah. So looking at this story, I would say, I mean, if we're, I don't know that, can we translate, maybe, could we translate VAF levels to when they arrived on the scene? Is that the question? I mean, you can infer, but yeah. Yeah, so if I was writing a storybook about this one, so the GATA2, I would say, was there to start with because of the fact that we had the information in the post-induction marrow, that it remained. That doesn't conclusively prove that it's germline, I will say that. But then I would agree with Mike that this SRSF2 probably came on the scene quite readily thereafter, that being involved in splicing. And then the driving with the NPM1 and the IDH1 those would probably be shortly thereafter. It looks as if the RUNX1 is new on the scene if we go by VAF alone.
Is it new or is it chip?
Well, I don't think it's chip once they've already gone to AM.
We don't know when the runners is running the other one.
Yeah, it could be separate. Yeah. Yeah, exactly. And the only way to really know that is to do single cell sequencing or some type of cell sorting mechanism up front.
Chip at the low level and
I think we can kind of infer a little bit more from this. We look at these VAFs and we're like, oh, the SRSF2 is 50%, so basically every single cell has it in one copy. And that's an assumption, assuming that you have two copies. That's
Yes.
50% to change the matter, right? So it may not be that every cell has one copy, might be only half the cells have mutated this.
Yeah.
What happens to me very briefly? Like, I would follow up these germline ones in the clinical lab now. What happened with the patient and the implications for their family? Let's say.
So for the clinical process to follow up on that is the individual in question has a skin biopsy done. So we take, not we, but the clinician does, takes the skin biopsy and sends it to the clinical lab. What we do is we do a culture. So we culture those fibroblasts, we extract the DNA. So that takes a couple weeks, 2 to 3 weeks usually. And then we do the sequencing only specific for that variant that we're questioning to be germline. And then if it is present in the skin biopsy, the assumption is that because we've now looked at a separate differentiated from cell development type of source, I say assumption because it's still, there can be other things here at play, but the assumption is that it is in germline, and then we can test family members from that point on. But if we're able to actually validate this, which we're which would be amazing. It would limit all that work. So all of the, like the patient wouldn't have to go and get a skin biopsy, which is one of the more beneficial things if they're already getting other testing. And we could tell from this testing that because all of their drivers are disappearing, they have this other germline susceptibility variant. Yeah, David.
Just note about the molecular evolution of GATA2 variants is, I mean, if you were going to do the family testing,
Yeah.
you
Well, it's like what we do now, right? With breast cancer familial testing as well. So you give them the information that they have this susceptibility variant. It doesn't mean even with breast cancer, it's not 100%. So there are still other environmental factors at play. It's just knowledge for individuals in the family. Generally, with all of this type of testing, you have to be an adult, unless there's certain exceptions. So when it's TP53 and Leishmania and things that can happen in a pediatric state, NF1, we do those in pediatric patients. But when it is something more adult onset, it's usually adult testing. Yeah.
Recognition in general of like somatic cancer testing, hereditary testing needs to kind of work in conjunction with together, and patients need to be informed. I think too, like only people that want to get tested will get tested.
For sure. Yeah. Yeah.
And also, pretty symptoms of cancer risk, certainly on RONX1, GATA2, there's increased risk of myeloid neoplasms. But the GATA2s are also linked to deficiency syndrome. Loss and K cells as well. And RONX1 from cytopenia and bleeding risk, and so there's more layers to this, right? It's not from the family who potentially have higher risk of myeloid neoplasia, but other things who aren't just in the plastic.
Yeah, great points.
I think we've had a nice, robust discussion. Thank you very much for stimulating that.